Hello viewers, welcome to the academia. Today I will discuss about rDNA technology. Genetic engineering primarily involves the manipulation of genetic material, that is DNA, to achieve the desired goal in a predetermined way. Some other terms are also in common use to describe genetic engineering, like gene manipulation, recombinant DNA technology, and gene cloning, gene modifications, new genetics. So these are the other terms. So recombinant DNA or rDNA technology is one of that. So if we see the history of the recombinant DNA technology, so the experiments performed by scientists Boyer and Cohen in 1973. In their experiments, they successfully recombined two plasmid, PSC101 and PSC102, and cloned the new plasmid in E. coli, which is a bacteria. Okay, so some biotechnologists who admire Boyer and Cohen experiments, they divide the subject into two chronological category, that is before Boyer and Cohen, BBC biotechnology, and after Boyer and Cohen, that is ABC biotechnology. So in the principle of rDNA technology, today I focus how it actually has done. So there are vectors used. We should have a donor DNA, that is our desired DNA we know we want, and the plasmid DNA also. So the type of vectors used, plasmids, that is 5 to 10 kb only, and PUC18, which is 2686 base pair. In host cell, it replicates 500 copies as per cell and contains several RD sites. So plasmids are one of the vectors. Okay. Next, bacteriophage, which are 10 to 15 kb. So lambda, which is able to replace 1/3 of the chromosome with large pieces of foreign DNA, and M13. Next, cosmids, which are 50 kb, hybrid vectors, part plasmid and part lambda, by which the cosmids are formed, and BACS and YACS, both are 300 kb up to 1000 kb. So these are the artificial chromosomes. So here you can see that the types, different types of vectors which are used. Okay. So in this slide we see the main steps involved in molecular cloning using recombinant technology. So number 1 is the DNA from donor is isolated and purified. Number 2, restriction enzymes. So basically endonucleases generate fragments of purified DNA by cutting the DNA at recognition site. There are over 100 restriction enzymes, each cutting in a very precise way a specific base sequence of the DNA molecule, not exceeding 4 to 6 BP. So the desired sequence we want, the restriction endonucleases cut at the specific base sequence. Okay, and next, the fragments are inserted, pasted, or spliced into plasmid. So this is called the technique, and the plasmid transferred to host cell. As the host cell replicates, recombinant molecules are passed on to progeny known as clones. Okay. Now cloned DNA can be recovered and analyzed from the host cell after processing. So we can take them and then we get our desired products. Basically, we clone it in bacteria. Next, cutting and ligation of fragmented DNA from donor and from the plasmid. So this is the detailed step, restriction enzyme action of EcoRI. So you can see the enzyme cuts both DNA strand at the same site. Okay. DNA fragments join at sticky ends And then the third one is the recombinant DNA. Okay, right? Next, the sticky ends from two different DNA molecules. Here we get a hybrid molecule, right? They hybridize together. Then the nicks are sealed using ligase enzyme. The result is rDNA. When this recombinant vector is inserted into E. coli, which is a bacteria, the cell possesses the instructions, and by translation and transcription and translation, it assembles the amino acid forming the protein product of interest, which we want. More importantly, the new instructions are passed along to the next generation of E. coli cells, forming recombinant clones on the culture agar. So here in the diagram you can see that how the insertion of DNA sample into a plasmid. One example is that the cloning of insulin A and B chains using rDNA technology. So number 1, DNA strand with the specific nucleotide sequence for insulin chain A and chain B. Number 2, unraveling strands of the DNA of chromosome 11 with the exposed nucleotides coding for the A and B chain of insulin. Number 3, restriction endonucleases, actually these are called natural seizures, cut the insulin gene fragment by recognizing the particular stretches of nucleotide, okay, and they are very specific. Number 4, the synthetic A and B chain genes, bacterial enzymes, beta galactosidase genes are separately inserted into the gene for a bacterial enzyme, beta galactosidase, which is spliced into and carried by the plasmid following complete compatibility. Okay. So you can see the E. coli plasmids. Now the recombinant plasmids are then introduced into E. coli cell. Practical use of recombinant DNA technology in the synthesis of human insulin requires millions of copies of the bacteria whose plasmid has been combined with the insulin gene in order to yield insulin. So if we want insulin commercially, then it is the way by which we can get it out. So this is very brief about the vectors of this process and the technology. If you have any query or question, you can ask or can comment in the comment box. Again, I will come about a different technique from molecular biology. Till then, good night.